Good afternoon, dear colleagues. The topic of my work is biosynthesis of cadmium sulfide quantum dots using Pleurotus ostreatus. I represent the Institute of Food Biotechnology and Genomics, National Academy of Sciences of Ukraine. And the second organization is Department of Physics, Jacq International University of Kyiv. First of all, it is necessary to explain what is a quantum dot. So a quantum dot is a fluorescent semiconductor nanocrystal which consists of the elements from the fourth, second, fourth or third, fifth group in the periodic system. For example, it can be cadmium sulfide, cadmium telluride, or indium phosphomide. They range in size from 2 to 10 nanometers and contain approximately from 200 to 10,000 atoms. Besides, it is interesting that semiconductor nanocrystals have interesting optical properties. Here you can see that nanocrystals' fluorescence color depends on their surface chemistry and size. Namely, large quantum dots have a red fluorescence spectrum and relatively small quantum dots have a blue fluorescence spectrum respectively. And now I want to draw your attention to the main aspects of biological synthesis of semiconductor nanoparticles. First of all, this green route of synthesis doesn't require toxic chemical agents, high temperatures and high pressure. Biological formation of nanoparticles is relatively fast, inexpensive, and it is not too energy intensive. And resulting nanomaterials are environmental friendly and safe for human health in low concentrations. So the purposes of our study are to obtain cadmium sulfide quantum dots using a culture liquid from the oyster fungus, Pleurotus ostreatus, to carry out physical analysis of their morphology, surface characteristics, and luminescent properties, and to demonstrate an efficiency of the fungus for obtaining quantum dots. It is important to note that the present study is the first effort devoted to obtaining cadmium sulfide quantum dots using the fungus Pleurotus ostreatus. This fungus is agaric, as you can see here. Pleurotus is agaric tree-destroying oyster mushroom that is widespread in many temperate and subtropical forests throughout the world. Besides, it is interesting that Pleurotus is valuable dye product, and in addition, it has antibacterial activity. So in this picture, you can see also cadmium sulfide formation procedure. So mycelium of the fungus was grown on glucose peptone yeast medium at the temperature 28 degrees during 10 days. Then we added inorganic salts such as cadmium sulfate and sodium sulfide. And the duration of the biosynthesis was 17 days, 17 days, it means that 10 days the mycelium was cultured with cadmium sulfate, and 7 days the fungus mycelium was cultured with sodium sulfide. Then the obtained solution was centrified, filtered, and obtained sample was further investigated. So our results. It was found that maximum in absorption spectrum corresponds to the wavelength. It is bad to see that, but the maximum corresponds to the wavelength 453 nanometers. And at the emission spectrum, we observed several distinct peaks at 431, 462, 486, and 528 nanometers. And these peaks were caused by the presence of the nanoparticles of different sizes in our sample. Evaluation of the size of cadmium sulfide nanoparticles from absorption spectrum was carried out by the empirical formula, which you can see at this slide. And we established in our experiment that the size of these semiconductor nanocrystals was in the range from 5 to 8 nanometers. Then, by the method of electron diffraction, we established that obtained quantum dots, cadmium sulfide quantum dots, have crystalline structure, and they correspond to the cadmium sulfide quantum dots. Cadmium sulfide quantum dots, at World-Seed modification. And besides, by the method of energy dispersive spectroscopy, the percentage of the elements in the visual field was identified. And as you can see here, the content of cadmium and sulfur was in the range from 20 to 25%. Besides, there were other elements in our sample, for example, oxide, silicon, and others. And their presence is caused by the biological activity of the fungus, and as well as the chemical components that were part of the culture medium. Conclusions. Basidiomycetes Pleurotus ostreatus is an effective biological capsitant for nanotechnological transformations. A spectral analysis of cadmium sulfide quantum dots shows that obtained absorption and emission peaks are typical for cadmium sulfide nanoparticles. Using the method of transmission electron microscopy, it was revealed that obtained quantum dots have spherical morphology and a diameter from 5 to 8 nanometers. And it is important that in the next step of our research we will focus on the study of the stabilization mechanisms of these semiconductor nanoparticles. Thank you for your attention.
Thank you. So we have plenty of time for questions. Yeah.
Thank you for your talk. As far as I understood, you have got your quantum dots together with the biomass of your fungus, and you did not separate them. So how do you intend to use these quantum dots? As far as I know, the separation process takes some time, and it is a challenging task to purify them from biological samples. This is the first question. So let us see the answer.
Yes, thank you. I just started to investigate this area, and I think it is my future work to try to separate these nanoparticles from biological capsitants. This work was the first study that in which we obtained stable quantum dots using this fungus mycelium.
So you hope that the separation process will not be that difficult, so you will be able to separate and purify them?
I think yes.
And there is some information on that in the literature from other sources, yeah?
About their mushrooms, there is no information. Our work is first. But
from other biological samples,
about separation?
Yes.
Yes, it is a bit
so
information.
It is an easy task. Okay. The second small question. So about the probable mechanism of the formation of cadmium sulfide quantum dots. If you, for example, if you take a filter paper, soak it with cadmium sulfide, and then soak it in a solution of sodium sulfide. wouldn't you be able to receive something like that without the living system like Pleurotus? Maybe it is a chemical process and it would be easier.
Yes, I understand. But the chemical process allow us to obtain large clusters of cadmium sulfide, and these aggregates are not quantum dots. Their size is not in nanometers range, nanometer scale. Yes, they are large clusters of these nanocrystals, and this biological method allowed us to obtain namely nanometer scale quantum dots.
And you think that the biological activity is the main point of obtaining these?
Yes.
Okay, thank you.
Okay, so maybe you had a question? So thanks again. And so